Oops. So, hello everyone. Sorry for the delay. We're setting up the
Okay.
So, hello everyone. Sorry for the delay. I'm just going to quickly share my screen and get started. Okay. Okay, so if somebody on the chat could actually confirm that you're able to hear me, we could get started.
Yes, it's all good.
Okay. So sorry for the delay. Seems like there are certain things more complicated than brain encoding. So thanks for those feedback. Okay, so we have two parts for this tutorial. The first part, I'm gonna talk about encoding models and present a tutorial on basic encoding. It's essentially gonna use the ideas that you've seen so far in this educational, using machine learning, scikit-learn, learn, that stuff in Python. And then after the break, we'll have the second part, which is advanced encoding, which will be given by my colleagues, Isil and Alexandre. So let's get started. So So what is encoding? Encoding generally means you try to transform something into a code. Okay, so in the context of brains, the code that we're interested in is what the brain represents. Okay, how we can measure that is, it's obviously neuronal activity. We measure that directly, indirectly, using different means, and that's the code. So this encoding analysis is a different paradigm of analysis is compared to traditional analysis, which I'll talk about in some time. But this essentially relies on this hypotheses or a belief of certain people that the brain represents something about its its stimuli, okay, its input. So, and these representations are transformed successively to make decisions, to perceive things, and so on, for other higher order cognition. So encoding, any encoding in general maps some input to a code, similar to like all these MP3, MP4, those formats and all that. So this is encoding. So so in the context of brains, this is like a prediction problem, and we need to predict these so-called brain responses from the stimuli or other attributes. So the output for this prediction problem is going to be the brain response as measured through some neuroimaging signal. And the input could be the stimuli in the form of audio, image, language, or some task condition, and so on. It could be behavioral attributes, button presses, answers to certain questions, and so on. Okay, or it could be any other kind of responses. So there, in this analysis framework of encoding, the requirement is that we frame this as a prediction problem of predicting brain responses. So this mapping from the input space to the brain response space helps us analyze these activities and which properties of the stimuli are encoded in the response. Okay, that's the framework to learn these associations. So what are these stimuli? We have a lot of variations in the stimuli that we use in cognitive tasks and what our brains are capable of experiencing and perceiving. So for example, we have text, image, video, audio, and so on. Raw stimuli usually have a very high number of dimensions. So, dimensions here is in the context of how the data is represented. And for example, an image could have like a bunch of pixels, and in order to have a good resolution image, you need to have many pixels. So that increases dimensionality, right? So each pixel contributes to a dimension or more, okay, depending on format. So some stimuli types like text do not have a natural representation. We need to use some methods to represent them. It can be incorporated into this prediction problem, right? It's a mathematical model, right, which maps from input to the output. So if it's just a bunch of characters or words, it needs to be transformed into some representation. So so we need an encoding framework which handles all these issues, representing the stimuli and mapping it to brain responses. So talking about the response, we need good pre-processing steps to actually ensure that the brain responses are these could be organized into modes in general, which could be like voxels, electrodes, Oops. Okay. I think the audio should work now. Yeah. Okay. So, yeah, it's perfect. Okay. Same. Okay. Okay. Thank you very much. So yeah, these are the questions we try to address using encoding models. Does the variation in the response depend on the stimulus? How well do the responses encode the stimuli? And how well are the responses explained by the stimuli, right? And can some responses be explained by specific stimuli? Do we have specific category encoding or stimuli type encoding in responses? And how can we quantify the dependence of responses on the stimuli? So these are some questions we'll try to address using the encoding framework, and we're going to look at the tutorial where we look at a specific example for this. So in traditional approaches of analysis of brain images, we are interested in similar questions, but we have a different approach. We use, for example, if you consider the problem of figuring out parts of the ventral visual stream, which are more responsive to faces, and the others which are more responsive to objects. Okay, so this is a classical problem you might have come across if you work with brain imaging, and there is some evidence based on studies that some parts of the visual ventral stream are more responsive to images of faces and others for objects. So what we typically do is we use like a difference in mean activity for these different task conditions, which is faces and objects, and we build statistical models of like with t-tests and all that. So so that's the traditional approach to establish that some parts of the brain are more selective towards faces and some of the others which are more selective towards objects. So this is the traditional setup. So in the case of encoding models, we can we're gonna we're gonna use a different approach. So theoretically, this approach, the traditional approach, is called a deductive approach, and the approach using encoding models is called an abductive approach. And also, you can use GLMs to encode different task conditions. You could have like a binary vector for like you could have a faces and objects as different categories, and faces is one, objects is zero, and objects is one, and faces is zero, and so on. You could have these categorical uh, information and try to use them in a GLM for analyzing. Okay, so in the encoding setup, we're trying to go from some input space to the response. So what are these inputs? What could these inputs possibly be? These could be these like semantic features, such as for example in this particular case, this could be an animate or an inanimate object, and we can encode these as feature vectors which represent these stimulus, and we could map them to these responses on the brain, and the reverse case is decoding, which is the other session that's happening. And we could also, yeah. Oops, sorry. Okay, I just missed my multitasking. Okay, so so yeah. So now there's also we could also encode behavioral responses, behavioral responses given by participants to brain responses this way. So that's another format in which you could use encoding models, like yes, no, unsure, these sort of questions. And then you could also use responses from some brain areas to encode responses in other brain areas. Okay, this sort of analysis is done in many work, and we could use some other neural variables to encode. And more recently, we're seeing an explosion of usage of artificial neural network derived features from stimulus and encoding brain responses from these ANN features. Okay, so uh, this essentially involves having some like stimuli encoding models, which is one type of a problem. We consider the stimuli and we extract some features based on the stimuli, and we map them to the responses of individual voxels. So we're gonna predict the responses of these individual units. If you consider functional imaging, this you could consider individual voxels, and we could try to decouple voxels which are more selective to different types of input. Okay, this way. So this is going to be our setup, and category encoding models use these categorical information about, for example, like a person, animal, car, vehicle, and we use these to map to these individual voxel or channel responses, and uh, we use these predictions to identify voxels which are selective. And we also have these feature encoding models, which was like I discussed these 4 cases a few slides back. Okay, so feature encoding models, which use complex features from stimuli for encoding, and uh, so these features can actually represent some natural statistics in the stimuli, or based on the domain of the stimuli, the nature of these features could vary. Uh, so these features may or may not be relevant to the brain. Okay, that's another thing. So features mapped to brain activity are like mostly use linear models for encoding. So why linear? That's the big question. So linear is like the simplest relationship between 2 quantities. So if you can predict one quantity from another using a simple linear mapping, then it means these two are very related, right? For example, you could consider the Celsius scale and the Fahrenheit scale. You could predict one from the other using a simple linear mapping. And although it's annoying sometimes if you go down south, they use another measurement system, but it's a simple linear relationship. They're very related, right? So that's the first motivation. And second thing is that it's more interpretable, uh, because you have coefficients for each dimension in the input feature. You can interpret like the relative strength of these different features, and you can identify like other insights from it. And linear relationship is a well-studied statistical framework. We have so many theories and statistical ideas around it. So we could employ these tools to better analyze brain data. And yeah, it works well with less data in the context of neuroimaging responses, where we don't have the luxury of having like tons and tons of data, as the machine learning community has. Uh, Linear learns better in low data regime, right? And also it's computationally less heavy. So this whole thing can be viewed from the perspective of the data, right? So if you trying to encode features from images to brain responses, you could look at this picture uh, where you have like the pixel space, right? Uh, so in general, in uh, if you have data, let's say images, we consider all the possible images. Let's consider you have like a 16 times 16, okay, so 256 pixels, okay, that image, and you have 256 possible values for each pixel in the image. So all these possible images exist in a certain space. Okay, we call that as a pixel space. So any image that you consider has a specific set of coordinates in this space, which represents the pixel values, and that's an image. So we try to map these pixel from the pixel space to the brain activity space. But this mapping happens in two steps. Like the first step is mapping the pixel space to the feature space. So this feature could be some any visual feature. You could just count the number of dogs in the picture, or you could like have a level of different colors in the picture. You could have like the distribution of these edges, horizontal, vertical, those edges in the picture. So any kind of features that you extract from the pixel space, because the pixel space is high in dimensionality, right? So a feature space is something that extracts more useful information from the high dimensional space. And you have a feature space, again, which has its own set of dimensions. And this is the feature mapping, okay? And once you have this feature space, you could map this to the activity space, which we refer to the brain activity. Depending on your modality, it could vary. So in general, our hypotheses about the feature space, which is normally a nonlinear transform from the pixel space to the feature space, that is mapped on to the brain responses using a linear transform. Okay. So they're just fixing. Yeah.
Yeah, I think it would be.
I'm gonna post the slides on Discord, just for following. Sorry for the interruption again.
J'ai entendu un petit clap ici. Ça a surchauffé.
Okay, it's on the brain encoding channel on the Discord. Okay. So this is this overall setup. You have some stimuli in some space. You extract features, and then this feature space is now mapped to the brain responses, typically using a linear transform, which is a regression. Okay. So so this can be viewed as a transformation between these spaces, for example, the pixel space and the feature space, using this nonlinear mapping, and using a linear mapping to the brain activity space. So you can picture this feature space, which is an intermediary between the input space and the brain activity space. And if this feature that you extracted from the input space is very brain relevant, then a simple linear mapping should help you associate these features with different brain areas. So that's the idea. So if you look at that particular slide, which has transformation between spaces title. Sorry. So I'm not able to see the number because I'm sharing, but yeah. So 15. Yeah, it's 15. Okay. So this is the picture. Starting with the stimulus, we go to a feature space, and then from the feature space, the brain activity space. And now you can see this brain activity space in fMRI. It's the voxel space. And how you can see this transformation happens from one space to the other. And this encoding helps us have a simple mapping between the feature space and the brain activity space. So there are some examples given. I'm going to stomp through them in the interest of time. In computer vision, you have some things called like Gabor filters, from which you can use to extract visual features from stimuli, and that's like a nonlinear mapping. And then from there, you can like map it to the responses of each voxel. And if you go to the method, okay, a few slides later, you will see that the overall setup actually has tell me the slide number. Okay, I'm gonna open this slide on the other thing. It's slide 19. Okay, thanks a lot, Isil. So if you go to slide 19, that's the mathematical framework for this. We're going to use a simple regression, right? It has to be linear, and a simple linear regression is not sufficient to map like a high dimensional like feature space to responses which are very less in number. If you recall from the lectures that from the sessions that you saw yesterday and this morning, you know, you this kind of a regression problem, or in general any learning problem where you have more dimensions than samples, right? When you have more dimensions, that it's an ill-posed problem. There's no mathematical guarantee that you will converge to a good set of weights parameters. So if you use ridge regression, it helps you deal with that issue. Ridge regression is just like an additional regularization to compress these weights, and that's something we're going to see. But overall, this is the setup. From the stimulus that is fed to the brain, you get these brain responses, and from the stimulus, you have some features extracted, and you have a transformation from the feature space to the responses using a linear mapping. So you can see in the next couple of slides that simple linear regression uses this notion of predicting, estimating the values of y, which is the output variable or the response variable, prediction, y hat. Okay, it tries to compare them with the actual y values. Okay, the y's without the hat, and the objective function which is used, the cost function that is used in the optimization, is the reduction of it tries to bring the predicted y, which is the y hat, closer to y as possible. So I told you this is very prone to overfitting, and there could be a lot of outliers in the data, and there's this problem of multicollinearity, where the features could themselves be dependent on each other. So the solution is using regularized linear regression, which uses like a constraint on the magnitude of these weights, the parameters, and we could use any of these like lasso, ridge, and elastic linear regressions, but ridge is used a lot in practice. So I'm just going to skip through the next few slides in the interest of time. But I'm just going to give you a gist of what's happening. So given that this is a simple linear regression problem, there are some issues that you could run into. The first thing is the number of samples in the input and the output data could be different, right? So if you have images, if you have like a movie which has like a frame rate of 24 frames per second, and so per second, the participant from whom you're recording the brain data is looking at 24 different images in a single second. But if you use like a signal such as a fMRI bold signal, you would have it depends on your TR. Okay, you could only sample one brain image in 1 second or a second and a half, and so on. So this creates an inequality between the number of samples in the input data and the number of samples in the output data. So we need some way to like associate the input to the output, and if the numbers are different, we need to kind of resample one of these two. Okay, depending on your problem, you could have like more samples in the output data than the input data, or you could have more samples in the input data than the output data. So you could either use downsampling to like just drop some frames in the input data, or in the case of EEG, MEG, you could drop some frames, or you could use like a smoothing function, and so on. And then you could have an HRF convolution in the feature sequence to resample the input data to match the rate of the output data. So that's in the next slide. You will see the illustration of the HRF convolution, which is normally used in like fMRI bold. So this is like a typical response for a for an event-based input, and you could convolve this to your with your actual input to better resample your input data. Okay. And there's also the second issue of delay. Okay, in the case of fMRI bold, so this delay problem is applicable to many other neuroimaging modalities, but in fMRI bold, it's very significant. How can we be sure that the output at time t is based on input at time t, right? So in fMRI bold, the output signal is a very slow signal, and the response does not really correspond to the input stimulus at the same time. Okay, so we could have different options of like shifting the output sequence by some number in order to like align the input to the output, because the input that you see at a certain time step, the response that is recorded is like slightly delayed. You could use an FIR model where you could put the time shifted features together as additional regressors, or you could use an HRF convolution. And finally, now that we saw how like the modifications that we need to do with this regression setup, there is this model evaluation you could do, you should do to see how well your encoding model has made predictions as compared to the actual responses in the brain. Okay, and we need to have a mechanism to score this quality of the prediction. So the error that we use normally is the mean squared error. As I told you, it's the individual difference between the predictions and the actual responses squared together and averaged. So this is a natural choice for measuring the quality of prediction, but it's sensitive to outliers, and it's not bounded, right? So error could be like 1,000, 10,000, 10, 1. It means less because it's based on the overall distribution. So that's where this could be useful. The R squared metric, which you can see on the next slide, which was also used in the model selection and validation session in the morning. R squared is typically used in this regression setup. It's a good choice for evaluation. It expresses how good the regression fit is relative to the mean baseline. Okay, if you were to not predict anything, just average all the Y's and give the average as a prediction, whatever how well you do is kind of the baseline for this. So how well is your prediction doing compared to this kind of mean output all the time? And this metric is not sensitive to outliers, and having a higher value of the R squared metric, it gives you a better fit. The R squared is also called the coefficient of determination, and it's a relative value as opposed to the MSE, the mean square error, which is an absolute value, and it helps to like assess the quality of the fit. So R squared typically ranges from 0 to 1, and it's a ratio which gives you like the share of a proportion of explained variance. Okay, what proportion of variance in X is explained in Y? Okay, so it ranges from 0 to 1, so you can easily picture the quality of a regression based on the value. And you can look at the math there. In the interest of time, I'm not getting into it. And I explained this. And if you go 2 slides further, you can see the different scenarios of the R squared metric. And you can see that if your prediction and if the actual values and the predictions of the responses match exactly, then you will, when you have a scatter plot of these two set of values, you will notice that they align on a straight line, and that corresponds to the scenario where the R squared value is 1. And the scenario where they're not even related, okay? So this is basic statistics when like your predictions and your actual responses are not even related, not even close, then your predictions are as good as random, and you will see like uh, the uh, the scatter plot forms like a circle, or like it drifts further away from the line, like x equals y line. So that corresponds to the scenario of r squared equals 0. And r squared, although it's a square, it could be negative. It just means that the predictions are worse than random. So if you have an r squared that is negative, it means the prediction of your model is worse than uh, randomly predicting something. So obviously, our ideal scenario is to get an r squared value close to 1, which means the regression fit is very good. And it's typically the range between 0 and 1. So you could use other metrics to gauge the quality of the regression, like Pearson's R, which is the correlation. R squared is just like a square of that conceptually. And you could use Spearman's correlation. You could use other second-order statistics, and you could use percentile scores, z-scores, and so on. And I'm just going to end with the last part of the pipeline, where once you do this fit and you have a way to score now, last step is to really ensure that the way in which you're scoring is valid and and it makes sense, right? So in validation and testing, uh, we need to ensure that the evaluation of the regression model needs to be done for truly unseen inputs. Because if you actually fit your model with a certain set of stimuli and you try to predict with the same stimuli, we call this thing being prone to overfitting, because it tries to like learn better for only those set of inputs, and it could not generalize, and this could perform well for that particular set of stimuli, but it might fail for other stimuli. So we need to ensure that we have this concept of truly unseen inputs, and we could potentially use cross validation methods such as k-fold, leave one out, and so on, to get a good estimate for the generalization of the prediction. Okay, because our whole modeling relies on the quality of the prediction in this regression model. We need to ensure that we have good checks on validation and testing. So we could also tune hyperparameters as part of the cross validation and stuff. So the next picture actually shows you the setup, where you have a training set. You should like set aside some part of the training set, some part of the data set as test set, and use the training set to train this kind of regression model. And finally, evaluate the scores like R squared, Pearson's R, and so on the test set, and assess the quality. So now in the tutorial, what we're going to do is we're going to like do a simple, very simple case of mapping uh, these to brain activity, and we're going to plot these individual scores, because our regression model is voxel wise. So individual voxels have like these are independent regression problems. So we're going to map these stimulus, the stimuli to the responses of individual voxels, and we're going to compute the score to assess the quality of prediction, and we're going to plot it on a brain map, and we're going to see if there is some association between the stimulus, the input data, and the responses. So I'm just going to leave the applications of encoding models to you, but I'm just going to quickly tell you that you could like use encoding models to like localize task relevant brain activity. You could see which parts of the brain are selective to different types of stimulus features and so on. Okay, and so again, like these brain maps correspond to like some kind of scoring of the quality of prediction in these encoding models, and you could determine which parts of the brain are selective. And next one you could understand this stimuli feature space. I showed you the concept of the stimuli, I mean the input space and the response space and so on, through some other spaces, right? The activity space, right? The input space and the activity space. The transformation happens through these different spaces in between, so you could understand these different spaces. And we're going to do this in the tutorial, but I don't think we'll have time. But anyway, we're going to see, we're going to identify receptive fields of brain areas. So there are some brain areas which respond to uh, uh, where there is like a field of response for inputs. So we could analyze receptive fields, and finally you could explain behavior using encoding models, and at last you could uncover representational hierarchies. Like you could see how the representations are transformed in different areas of the brain uh, with encoding models, and also reconstruct stimuli. Um, this is a decoding problem, but you could invert an encoding model using Bayesian statistics and uh, reconstruct inputs. Okay, so I'm gonna quickly share my screen with uh,
Shall I try? Maybe this is
with the tutorial. Um,
they don't respond. They maybe yes. Because it's less. Maybe. But I go to a spot. I want to even. What to know that? It's sharp.
okay, so all of you here, you have the link right from the tutorial. So you could either download the IPython notebook and launch it locally, or you could upload it to uh, if you install the stuff. If not, I would recommend that you upload it to Google Colab. But you need to like pip install Nylearn. I think that's the only dependency. So so I'm going to quickly summarize what we're going to do here, so that if we might not have time to do it fully, but uh, I'm just going to give you the context. So we're going to consider this particular study from Miyawaki et al., where they use like very simple images. They showed these visual stimuli to participants, and they collected data, fMRI bold data. And these images are 10 times 10, okay, just 100 pixels. They're like checkerboards, like 10 rows and 10 columns. And the image pixels are very simple. They're either black or white. So it's either 0 or 1. Okay, so so these images were shown to participants, and the brain activity was collected. So we're going to try and build a simple encoding model from these images to the brain responses in like visual areas of the brain, and we're going to see uh, we're going to analyze this. So before we jump into building our encoding models, obviously we need to set up our data. And if you look at cell number 1, basic imports, we're just importing NumPy, Matplotlib, NumPy for like array operations, and Matplotlib for plotting, and so on. And finally, and then the first step now involves uh, getting the data. So the data is available as part of Nylearn, and you just have to like call fetch Miyawaki 2008, and it takes a while to download, but it's very quick. It's a very small data set. Okay, download it. So once you download it, you can actually print the variable data set, which consists of the downloaded data. So it consists of the path. Okay, you can see it's like a dictionary, Python dictionary. You can see that the dictionary contains different values, keys, and values. So you can see there is a func key, which consists of path to figure run 1, figure run 2, run 3, and so on for 20 runs. And then you have label. You have run 1 label, run 2 label, and so on for 20 runs as CSV files. Okay, and then you have mask, um, which is the brain mask we apply on the data. And then you have these mask ROIs for different regions of interest, like left hemisphere, right hemisphere, the visual areas like V1, V2, V3, V4, and so on. Okay, and you have some information about the study. So I hope there is no question. There are no questions about the data as such so far. So now we need to so this contains a bunch of things, which is like yeah. Yeah. Yes. Okay, so we're back. Sorry for the trouble. It was the internet this time, having its fair share. So, yeah. So so the training data actually starts after the first 12 files. So we just have to like reindex things. So if you see there, we're going to consider random runs because there are two kinds of images which are used in the study. One set which are random images, which we'll see in some time. It's like a bunch of random pixels, black and white pixels. And then the other set are like shapes. You have like shapes like plus, cross, square, and so on, which are like regular like images. So we're going to consider the random images first. And so the index is like the file paths are after like the index 12, starting from index 12. So the fMRI, so we're concerned with two different kinds of streams of data. So the first one is uh, the the files of the brain activity, which is the fMRI random runs file names, and second thing is the links to the files to the stimulus. So those are the CSV files you saw. Uh, so fMRI random runs select starting from index 12 in func, and label consists of all these. If you remember these CSV file names. Yeah, so you can see those file paths here. So we're just collecting these paths in these variables. And now, if you look at the next section where we're pre-processing, masking, and extracting the data, we're using this multi-nifty masker to load the fMRI data, clean, and apply the mask. So the multi-nifty masker, if you just have to pass the mask file, which is part of our dataset itself, and so you can like have these extra properties to like detrend pre-processing steps like detrending and standardizing. Standardizing because the range of these bold responses could vary. Standardizing just like, subtracts the mean and divides it by the standard deviation to compress them into a range. And detrending, it's a common pre-processing step for bold fMRI bold data to like remove some global trends in the signal. Okay. So now once we have created the masker, we apply masker.fit, which you might have done yesterday if you attended the NeuroAI tutorial. And then after you fit the masker, the properties are set. Now we need to transform by passing the paths of the fMRI data. Okay, the paths of the fMRI data are in the fMRI random runs file names variable that we collected in the previous cell. And we have the fMRI data after applying the mask, detrending, standardizing, in this variable fMRI data. So our fMRI data is going to be the source of a response until now, after now. Okay. And what is the dimension of the fMRI data? It's going to be it's two-dimensional. It's going to be the number of samples times the number of voxels. Okay. So if you recall from yesterday, masking basically converts the data with a mask into like a two-dimensional array. You can consider each of these voxels to be on one axis, and for each voxel you have these different samples. Okay, the time course, the values of the time course. Okay, so the dimensions of the mass data matrix is like number of samples and number of voxels. You can print them out. You can like print the number of runs in the fMRI data is the length of the fMRI data. Okay, we have 20 runs, right? So we collected 20 runs. Don't remember 20 runs. So among these 20 runs, the dimensions of the fMRI data are in each run. Okay, fMRI data of 0 will give you run 0. Like basically it will give you run 1, but computer scientists count from 0, so it goes from run 0, fMRI data of 0 until fMRI data of 19. So shape. When we deal with NumPy arrays, we're interested in the shape of the array. So like Ed Sheeran. So we're going to use dot shape attribute for these variables. So fMRI data of 0 dot shape will give the shape of the run 0 data and so on. So we're just printing out the shape of run 0 data. And you can see the number of samples is 145 and the number of voxels is 5,438. So we have like data of around 5,000 voxels, which runs for 145 steps, okay, time course. So we're going to just print out the time course in the next cell. If you look at the image here, so in this cell, you can actually change the runs you want and the voxels for the corresponding voxels of these runs. Voxels should be between 0 and 5,437, right? Because you start from 0. And the runs should be between 0 and 144. So you could like switch these values in this cell, and you can actually run them. And I'm just going to check if I ran things. Okay, so if you do that, you'll observe the fMRI time courses this way. You will see like the y-axis is the normalized bold response. You can see it hovers around 0 because we've standardized it, and you can see that the time course goes for until like 145 time steps. And you pick the voxel, you have this time course. So this is the response. Okay, any questions so far with the response? How the response looks? Yeah.
So did it delete any voxels, or are the voxels?
So the 5,400, like those are the voxels. After applying the masker, it just like grabbed 5,400 and odd voxels.
There was like the mask itself is like 5,000.
Yeah. So these 5,400 voxels correspond to like the visual areas in the brain, which involve like many different areas. But because this is a visual task, we're just gonna, and also because it's a tutorial, we want to minimize the number of voxels. So we're concerned with the voxels which we know belong to the visual areas. Okay. But okay, this is the data, 145 time steps and so many voxels. So that's the idea. And the second part, so now that we've extracted, pre-processed, done everything with the response data, the activity data, let's now move on to the stimuli data in the next section. So we know the stimulus is shaped 10 times 10. Okay, it's like a square, and the data of the stimulus is stored in these CSV files. CSV files is just like your spreadsheet, okay, Excel sheet. You can see that it's 10 times 10. These values I told you are either black or white, and in this particular case, it corresponds to -1 and 1. So if it's black, it's -1. If it's white, it's 1. So you will see like the CSV files are structured as like a checkerboard, like 10 times 10, and these you can see these values there. So we're just going to use we're going to reshape. We're going to reshape. Actually, sorry. The CSV files actually contain like 100 values as a straight line, right? We call that operation as flattening, because instead of having a square, it's just gonna like consider each row like a strip and then stack them together horizontally. So we're just reshaping it in order to visualize it. So we're just reshaping. You can see the reshape here, reshape and load text from stimulus run and delimited by comma, because it's a CSV file. Each of these values are separated by a comma. And and you can see now that these the number of runs in the stimuli data are 20, and the dimensions of the stimuli data is actually number of samples times image height times image width. So it's going to be 145 times 10 times 10, because we have 145 time steps. In each of these time steps, one image, which is 10 times 10, was shown. That's the idea. Okay. And now you can see in the next part, we're just printing out the images. And before we print out, we just we're just using like the matplotlib function called imshow, image show, and you can look at these images yourself. Like you can change again the number of the run and the trial. So these individual like 145 time steps, we're calling them trials because different images were shown, right? So you can change the run number and the trial number, and you can see the kind of images that were shown. Okay, usually in trial 27, you can see all of them are black. In trial 125, you can see it's like, these are basically like your QR codes, right? Some random black and white patterns which were shown to the participants. Okay, and so creating the input and target matrices for training. So now we looked at the input data and the output data, and when we create the input and target matrices for training, we have to remember I told you about like some issues with like the hemodynamic delay in the data and things like sampling rate and so on. Okay, so here we see that there are 100 for a given run. We have 145 10 cross 10 images, and we have 145 responses in the fMRI data. So there's not so much of a like an association problem. Like we have a one-on-one correspondence between the input and the response. Okay, that's not an issue, but we should potentially handle the delay. So because fMRI bowl data is a very slow signal, we could potentially, so the response in the brain for an image which was shown at times t could only be observed at time t+1 or t+2 or t+3 or so on. Okay, that delay, there are people who study the delay and so on, but for the sake of simplicity, we could just fix that number delay. We could just say 2 steps. So the input that was shown at time t, response you get during time t+2. Okay, so that's the idea. So that's why you can see delay trials is set to 2. And what we're going to do is like just, this is we're kind of shifting. We're just shifting the data in this loop, and we're going to use this NumPy function called vstack, which is vertical stacking. We're just vertically stacking the delayed data and putting it together. And in doing this, what happens is we have to like get rid of something, right? Because the responses of the last two trials, we might not see it, right? Remember, we see the response at time input at time t. We see the response at time t plus 2. So for like 144th trial, we should see it at time 146, which we don't have data for. So we just crop out the data of the first two trials, and we crop out the data, the stimulus of the was there? Oh yeah, we crop out the data of the last two trials, and so we have 143 right now. Okay, so if you multiply 143 by 26, you will get this number 2,860. Okay, so so now our stimulus data is like 2860 times 10 times 10. So we have 2,860 images of 10 cross 10 images, arrays of 10 cross 10 images, and fMRI data is like 2,860. So many trials. You have like 5,438, so many voxel values. So now we have the input and the output data ready, except one thing. We want to do regression, and the input data has an additional dimension, right? It's 2,860, 10, 10. So so we need to just flatten it again. So these values, 10, 10, we're going to flatten it again and convert it to a 100-dimensional vector. So 10 cross 10 array is now converted to a 100-dimensional vector. And we're going to use the reshape function again. Okay, reshape. Now you can see that the stimuli data shape is 2,860, 100. Okay, so now we are going to map 2,860 100-dimensional vectors to 2,860 5438 values. Okay, and we're going to apply these for individual voxels. So for each, we're going to have 5,438 regression models to map these individual 100 dimensional vectors to these responses. Okay, so we talked briefly about the ridge regression models. We're going to use a voxel-wise encoding model for each of these voxels. We're going to try to establish this regression setup. We're going to use the ridge model from scikit-learn. Okay, you can click on it. You can read the documentation. Okay, I'm going to share my screen again. Okay, so now we have our input data and output data ready for the regression setup. So then we're going to use the sklearn linear model ridge, and we're going to use k-fold cross-validation for the evaluation. So we're going to use a 10-fold cross-validation. We're going to partition the data into 10 different folds. If you've attended like the morning session, you'd know. And we're going to fit ridge regressions for the remaining 9 folds and test it on the 10th fold. And we're going to like rotate the fold which gets to be in the test data. Okay, yeah, sorry. Okay. Yeah, so if you look at this loop here, we're just splitting the data into 10 different folds first, and then we're just looping through these folds to you can see we create a model called ridge with alpha, which is the parameter for hyperparameter for the ridge regression, the alpha value, which is set to 100. And you can see that the ridge model on the training data, you just have to call fit stimuli data of train and fMRI data of train. The train is just going to give us a list of index values which are selected for this particular fold based on the split method. And you can see that the stimuli data of the train set is fit with the fMRI data of the train set. And then once you have the fit model fit done, the learning is complete, and you have to just predict for the stimuli data of the test. So this is what I told was important. You train on the train data and you predict on the test data. And then once you have these predictions, you just add these predictions to the score. Okay, the score is just the R2 score between fMRI data and the predictions, fMRI data of the test set and the predictions that you obtain using the stimuli data of test sets with the trained model. And once you have these values, you can see for 10 folds, it considers like about 2,574 samples for the train and 286 samples for the test. If you add these together, you'll get that magic number 2,860 that you saw above. So finally, we're going to analyze the predictions, and we'll be done with this part of the tutorial. So to analyze the predictions, we have all these scores that we require in the scores variable. I told you that's the quality of the prediction of the regression model. And now you can actually, so because we have the so to analyze the predictions, we have like the predicted signals for the last fold of the k fold, okay, which is in the predictions variable because the loop runs one after the other. The last fold's result is actually there in the predictions, and we're just going to use that for the sake of simplicity. And we're gonna take the index values from the actual fMRI data, which corresponds to the original data, and we will just try to visualize them. Okay, and if you look at this, the actual data, you can just select like fMRI of test of if until like the half the length of the test data, and again predictions for the same index values. Only then the actual and the predicted value would match and would make sense to be corresponded. And again, we could just plot these two together because for each input it has the value of the prediction, and we can generate this kind of a time course using the predicted data, just like we have the actual data. And you can look at the plot below. The orange curve represents the predicted signal, and the blue curve represents the actual signal. And these are some good predictions. Like the first one, you can see they kind of match very closely. Okay, for voxel number 1951, and then for the next voxel, you will see that it kind of matches when the magnitude is lower, but for the peaks are not like matching. And the last one, you can see that it's completely not matching. So any thoughts about the predictions here? Do you see some characteristics of the prediction as compared to the actual signal? Do you have any stark observations? Yeah.
Flat.
Yeah, they're flat, right? So they're not as pointy as the actual signal. They have this kind of a boxcar type, and they're capped at some point. They're not able to go beyond a certain point.
But is that because of regression, or is it
Potentially, right? It doesn't overestimate, and like because it tends to not overfit. So it tries to take the safer route. So, and also you can see that just like bold signal, it has this kind of a like what we call as the refractory period, and okay, not so much refractory period, but then it just like falls flat. It's not like spiking all the time. It goes up and then falls flat, goes up and then falls flat. So it is responsive to some extent. So, and these are like the good, like the first one is like one of the best voxels. Okay, but the last one is like completely wrong. So is this a good thing or a bad thing? Do you want like all voxels to be predicted very well? No, right? We wanted different voxels to respond differently. Like some voxels are easier to predict based on the features you considered, and you can associate those features with these voxels. Some other voxels don't really care about the features that you used. So you could know that these voxels care less about the features that you use. So this difference is something which is useful for us rather than uh, like we don't want to achieve like uh, state of the art performance in all voxels as far as encoding analysis is concerned. Okay, so you can actually have a scatter plot of the predicted and the actual values this way. And I told you, like, the straight line, x equals y straight line, you can see that it's not perfect, but it's like kind of clustering around the line. And can you observe and tell me there's like a cluster of points in the top, and there's like a straight line of points at the bottom. Can you guess what like what these two different set of points could be on this time course? Okay. Okay. In the interest of time, I'm just going to tell you. So, the things that match are like the ones. So these areas where we have uh, the values actually having a closer match. But the things at the bottom are these. If you look at the values, look at the points where the prediction is low. Okay, the actual signal goes on the negative side, but the prediction does not go worse than some level. Okay, it's kind of flat at the bottom. So all these flattened values are like uh, they form these bad predictions at the bottom. Okay, so finally, we can plot this on a map. We can actually find the score, the R squared value of the best voxel, okay, not the best voxel, like voxel 1951. You can see that the Pearson R is 0.8 and the R2 value is 0.65. You can see there's a square relationship already, right? And so that's the value which we're going to plot on the brain. And in order to prepare the map to plot on the brain, we have the scores variable which consists of all the R squared values on the uh, masked structure. So we just have to like reject those values. We drop the R squared values which are less than 0 because they're not very useful for the analysis, and then we use this masker's inverse transform to go from the masked data structure to the brain volume again. And then after that, you can threshold based on some value. Here we're just using like a very small value, like 10 to the negative 1 to the negative 6. And yeah, so the score map is now of size 5438, the exact number of voxels we started with. And we can find the best voxel. Best voxel is voxel number 1951 with score 0.6. So this is just next cell is just to show you how you can like find the voxels which are like more which have like higher R squared value. You can see 0.6. There are about 3 voxels, 0.58, and so on. And you can look at the histogram of voxels. You can see most R squared values are 0, but there are some R squared value, like as the R squared value increases, that like you have lesser and lesser number of values. This is typically what you observe in voxel-wise encoding models. And to plot the values on the brain map, we have the brain image ready from the R squared values. You just have to like, we just have a function to map it to a specific location. And you can see this is the brain map with the predicted values. And so we have the R squared value. You can see some areas are yellow, which means they're very high, and some areas are red, which is medium. Some areas are black, which means R squared is 0. So what do you think it means? What do you think like the values in yellow actually mean? the areas in yellow. Yeah, they are the best predicted areas. And if you're if you're in neuroscience, can you guess what that area is in yellow? Cortex. I mean, all these areas are like, it's in the occipital, so it's all these are kind of visual, but the yellow area is okay. You can, I'm skipping in the interest of time. You can read below. I've noted it down. So these correspond to the like more primary areas of the visual stream. So because these are very basic, we use raw features, right? We do not do any feature extraction. We just use the raw features. These correspond to like what the V1 observes and like early visual areas observe, as opposed to like later areas. So you can see there is a higher association with early visual areas with these features. Okay. No, so it may yeah, the features, it's not about the pictures per se, because the features we use for this regression model, these are like raw features, pixel present or not, like pixel white or black. Okay, so that's something which is more correlated with the responses of like early visual areas, as opposed to later areas which are more high level. Okay, so the features map to these responses, and now you can actually find like areas which are more responsive, less responsive. Like, the black voxels here, they don't really care about, they don't seem to care about the features that we use. Okay, that gives you some insight. So this is like a analysis from an analysis point of view. Like, as I told you, if you look at the traditional analysis, you would use different conditions and like statistical maps based on these differences, t-tests and all that. This is a different way of analyzing them. That's the main message that I wish to deliver today. But I will like stop for a break, and then we'll continue the next section. Or yeah, before the break, you just have to install this. But there is this next section on the receptive fields, which I'll invite you to like do it later at your own time. I feel this is very interesting. You can actually associate which voxels in the brain are responsive to which pixels in the image. Okay, you can see like some, if you look at this analysis, you can see that you can see the positions of these voxels. Like some voxels, like if you consider a particular voxel on the in a position, you will see some voxels which correspond to the particular pixel in the image. You can see some voxels which respond to the left, the top, to the right, to the bottom of the image. And it's unfortunately, because of time, I'm not able to go through them, because the other tutorial is also very interesting. I feel like I don't want to eat into their time, and also it's a Saturday evening. So yeah, so I'm around if you have any other questions, and I'm just gonna let my colleague Alexandre continue for installation instructions before you go to the break. Thank you. I'm gonna add you.
So that's definitely if the lamp is died, or maybe we can try.
Just before taking the pause, if you not already have done it, just clone the repo that is being shared on the slides, and install the required package, and then open the notebook. And once it's done, you will run you will have to fill a small form in which you will give us the link to download the fMRI data. And then you have to put it in the. So once you open the notebook, you have to fill the link, fill the string with the link to the fMRI data in one of the cells it's indicated. Then say that you want to download the data. Can you see my screen on the? No, you don't see my screen.
Where is this form?
Share
Can
screen.
you show me where the form is?
Yeah, but it was the case. Doesn't seem to work.
Is that
C'est le même fenêtre.
Je peux pas le faire sur tout l'écran,
Oui, tu peux partager. Tu vas switcher.
mais ça marche pas.
C'est quel écran? Ok, ça marche. Normalement, ok. Onglet Chrome, c'est quel onglet?
C'est donc je peux pas partager.
A priori, ça marche.
Onglet marche normalement.
Je peux pas partager ça.
C'est où?
Je peux pas partager.
Tu peux
les
juste
autres.
choisir, c'est sur le Safari?
Non, c'est du code.
Non, c'est quelle application?
Code.
Oh, c'est code. Ok, il faut choisir cette fenêtre.
Ok.
Mais pour l'instant, tu vas. Je vais fixer cette situation, mais pour l'instant, tu vas partager ça.
Ouais.
Tu as donné les instructions?
Soit là,
Il y a
en
plus
fait,
personne,
tu
de
vas
toute façon.
regarder sur le manuel et en
Ok.
fait, sur le manuel, ils
Donc,
disent, si
ok,
c'est rouge, ça veut dire
est-ce que
soit
je peux te
c'est genre à
muter?
moins de 5% et que c'est la lampe est quasi grillée. Soit ils disent rebrancher.
Okay. Okay.
No one responded.
You're audible. You're on the screen visible. Okay, we can start. We can continue.
Can you can you get
We need to
the
mute this one. We need to mute this one. Mute tab. Okay.
Okay, perfect. So hello everyone. Now we'll follow with a bit more advanced encoding. So I hope you all have cloned the repository that was indicated. You can find it here on GitHub. Then you just have to install the requirements and open Jupyter Notebook to open the notebook which is in the main repository. If you have any issue, you can ask Pravish or Isil, who might help you to do it. Do not hesitate, or if you have really big issues, don't hesitate to follow with your neighbors. Okay, so with Pravish, you've seen that in order to study brain data, when you use encoding model, you could, sorry, it's a bit slow, you could manually derive some representation from a stimuli. So for example, given an image of a dog, given a set of images, and you want to probe some activation in the visual system in the brain, you might want to find brain regions that are involved in the processing of faces, or human, or animals, or objects. And in order to do so, you could manually encode the presence or the absence in a given picture of this kind of stimuli. For example, in every picture that had a human, you will have put the predictor just a 1, and when there is no human, you will put a 0. So this is clearly a basic regressor that you can use in some encoding paradigms. But the default with this, the issue with this kind of predictors is that it's quite a poor representation of your stimuli, and you might lose a lot of information that is being encoded in your image or in your other kind of stimuli if you use a text input or audio input, auditory input. So the idea is that you want to build the more complete, richer representation in order to capture all the variants that you can in your brain data. And to do so, it's impossible to do it manually, as there are too many dimensions in any kind of stimuli, whether it is visual, auditory, or textual. So this is why researchers have come with the idea of using machine learning models to build for themselves this kind of complex high-dimensional representations. And so the richer your representation is, the more fMRI signals, the more brain signal you will explain. So in this presentation, we'll focus and we'll work with fMRI brain data, and we will use some textual stimuli, meaning some text that is being read by the subject. And so the underlying hypothesis behind all that is that if you have a model from which you derive some representation, and your model explains very well some signal in a given brain region, then there is a high probability that the information that is processed by this brain region and the information that is being processed by your model are quite similar. So if you know what your model is doing, you can learn about what the brain region that are very well fitted are doing. And conversely, if you know what the brain region are doing, if you know what the brain region that are very well fitted by your model are doing, then you can infer what your model has learned, what your model, what kind of information your model is processing. And so there are there are different kind of stimuli that you can encounter, like previously said. You could have some visual stimuli, so you could experience some photos, some pictures, some videos, and there are different kind of models associated with each kind of stimuli. And each of these models is specialized to process this kind of information. For example, you can find that UNet model or ResNet or other kind of model like that will take as input a picture of or set of a sequence of picture, which is a movie, and then they will convert this stimuli into some vectorial representation. They will process it, and they will learn some kind of information about it. You can find some models that are used to process auditory stimuli, like a wave. For example, you're just listening to a story in the fMRI machine. Or you could also use some models that process some textual information. So you might have heard about transformer models that are that are state-of-the-art and have achieved quite impressive results. Recently, there has been the ChatGPT with which you can just ask question, talk, and it will give you super constructed and super coherent answer. And so each of these models learn to process some kind of stimuli and to build some representation for the different elements in your stimuli. For example, transformers, which is the kind of architecture that you see on the right, will create a vectorized representation, a vector, for each word that is being fed into the model. This representation encodes a lot of information. It encodes semantic information about the word, which is the meaning of the word. It encodes syntactic and contextual information, meaning how does your word interact with the other words that are in the context. And one advantage of this kind of state-of-the-art model is that they are, like I previously said, they're contextual, meaning that the representation that they build for each word depend on the words that occurred before. And basically, in French, the word plus can be thought as more or as no more. So, sorry? No longer. Yes, thank you. And so, if you don't have the context, you cannot infer the meaning of the word. And this is why these contextual models are really useful. So now we will start the processing of the notebook, but we'll start loading and preprocessing the data. I'm gonna share the other screen. Pravish. Yeah, it's here. Okay, you should be able to see in a few seconds my screen. Okay, so once you should have cloned the repository, installed the package, the requirements. Is it okay for everyone?
It's still downloading for me. The fMRI data, because I just started it like 10 minutes ago.
Okay. A bit more?
Yeah.
Okay, I'll go through go see everyone to see. And so the problem. Où est-ce que tu l'as fait?
C'est du data. Est-ce que tu quelque chose en main?
Là, tu es ici. Oui, il faut que tu sois dedans.
Si j'ai fait les deux, mais les deux étant.
Il faut que tu sois dans le. Pour ça, tu l'as fait en étant dans le même tutoriel.
Oh, ça sent ouais.
Ok, c'est bon, hein. Il y a eu un problème?
En fait, je
C'est bon, tout le monde. Main tutorial, c'est le nom du repo. Ouais, sur l'espace. C'est de télécharger les impératifs. T'as pas mis au bon endroit, t'as mis les CSV, mais tu l'as pas mis dans le dataset. Tu l'as remis?
Oui, parce que j'avais.
Voilà. Là, c'est bon.
En fait, j'ai
C'est bon, toi?
Mais en fait, moi j'ai fait.
So I'll keep going. So once you downloaded your fMRI data and your stimuli data, you can visualize it with the next function. So there is quite a lot of code behind it in order to have pretty surface projection. So you're working with volumic images, meaning that every image of the brain is a cube, and this big cube is segmented into smaller cube called voxels, and this represents the entire brain. And so in order to visualize it like that, you need to project every value on the cortex. So imagine a real brain. It's like taking every value at a certain distance from the surface and try to project it onto the very surface, and then you can visualize it like that. So you have the lateral face, left lateral face on the left, the left medial face. So imagine you have your two hemispheres. You're visualizing the interior parts of the left hemisphere here, the second one. The third one is the medial right hemisphere, and the last one is the lateral right hemisphere. It should take about 1 minute to compute for two images. So then in order to compute things in a relatively small amount of time, we just select the three first runs. So it's approximately 10 minutes for each file. And we are using the friends data. So these are subjects who have watched one episode of Friends, several episodes of Friends of several seasons. But here we are only working with 3 times 10 minutes of movie watching. So this is a naturalistic stimuli, meaning that you are put into condition that you might encounter in your everyday life. So it's then like we said before, to capture the richness of this kind of stimuli, we use these complex machine learning models in order to extract some information. So with this kind of stimuli, you have the visual feed, but you also have the subtitles, which bring some textual information to the subject. And so what we do here in this experiment is we will work only with the textual information. So we will use neural language processing models in order to extract some representation from the subtitles. And we use this representation to predict fMRI brain activity. Another thing that we could have done would be to use the image feed in order to give it to some visual processing models and use the representation learned by this model to feed brain data. So then we will process the fMRI brain data by creating a masker. So if you remember, this is the object that Pravish presented you a few minutes before. What it does is, given a sequence of fMRI data, it will look at the brain region where you have a useful signal. So useful hiding a lot of things behind it, but basically it's where you have a consistent signal across all the images. Then it will identify this brain region, these voxels, where you want to retrieve the signal, and it will go find all the time series of these voxels, and then we will use this time series in our encoding models, meaning these are the time series that we want to predict, and we don't want to predict or to focus or to grant some times to the voxels that are not in this mask. So we extract. You can run it at the same time. It should work if you have installed all the requirements and downloaded all the fMRI and the stimuli data. So we preprocess the fMRI data, meaning we just apply the masker to the fMRI data. Behind this function is another small thing, because in encoding model, when you're doing encoding, it's like every scientific experiment, there are a lot of things that are hidden because of some small issue that are going to crash everything. For example, if you have a voxel in brain data where there is signal, but the time course is constant, then in the end, it's super easy to predict this voxel. But when you're computing some metrics, like Pravish presented you, like the Pearson correlation or the coefficient of determination, you will have non-computable values, non-values, because the time series is constant. So these are small details that you want to avoid. So in order to avoid this kind of thing, we added a really little bit of noise to this voxel. So it's just a small detail, you can forget about it. Just to tell you that this kind of function can implement more complex preprocessing to your data. For the stimuli data, we just load the CSV file. And so these stimuli data are an MP array with 3 columns. One for the words that appears. So you have one word per row. And these are the words that appear in the subtitles. And for each word, you have the onset, which is a timestamp of when the beginning of the word occur, and the offset, which is the timestamp when the word finished. And then we plotted the time course of four voxels, as you can see. So you have 3 files of fMRI data. For each of these files, you have like almost 450 to 500 scans, so images, with 26,000 voxels, so which is quite a lot. The stimuli files look like that, so I describe it orally, but so you have the subtitles, the onsets, and the offsets. Okay, now I will go back to the presentation. Sorry, it's a bit difficult to switch from one to the other. So now we have loaded our data. We have preprocessed it. If you have an issue with the code, don't hesitate to ask for. And there are a lot of questions that you might want to consider when using some really complex tools like machine learning models to study brain data. For example, given my stimuli, which model should I use? And even if I want to process some textual information, you might want to consider several models that will have their own properties. And each model has its own specificity, like an architecture, a given number of layers. Each layer has its own embedding dimension. There are several operations that are being implemented in the models. So what is the impact of this kind of choice of architecture? There is the data that the model is trained on. So how much data did we use to train this model? So this has quite an impact on the performance of the encoding performance of your models. What was the nature of the data? For example, if you train your model on some Wikipedia pages, and then you use it to encode the brain activation when someone listens to some children's stories. So Wikipedia and children's stories are quite semantically far away from one another, and so you might have quite poor encoding performances. So you might want to choose a training data set that is close to the data that is being experimented by the subjects. Then there are more complex parameters, like the task the model was trained on. For example, language model can be trained to predict the next word. So given a few set of words, the dog runs, the model might be trained to find the most probable word that will occur after, so in the courtyard or in the house, and so on. It could be trained to do some denoising, to find a word that is being masked in a sentence. For example, given a sentence, you will mask one of the words, and you will try to find what word was it, depending on the words that are before and after the mask token. And then there is the objective function that is that the model used during learning, during training. And so basically, all these choices are quite important in order to understand which model to use and what you will be able to show with this model. And for example, if you have a visual stimuli and you want to use some CNN and a model with a CNN architecture, it has been shown, for example, that this model learns some hierarchical representation of the information. For example, the lowest layers will capture some Gabor filters information, meaning some really low-level information. Then they might capture some corners in your images, then some higher-level properties like a nose, an eye, a finger, and then the deepest layer might eventually be able to encode some information about faces, about objects, semantic, and so on. When you work with auditory stimuli, the lowest layer might capture like phonemes information, then morphemes information, then word level information, then sentence level information, and so on. So you see that with some models, you can have a hierarchical representation of the stimuli, and therefore, depending on what you want to show, what you want to do with your brain data, you might want to select the output of one layer compared to another. For text representation, it's a bit more difficult because transformers do not have this hierarchical representation of the input. In fact, it's more of a continuous transformation of your input, meaning that you have several layers, and the first layers just make a guess for the task it was trained on, and then even if it's not the right guess, then it goes through the second layer, which also tries to make a guess, and so on. And so this is a kind of continuous refinement of its prediction. Okay, So now, like described before, we will try to use the textual information displayed during the frame movies in order to predict the fMRI brain activation. So we will load the models, and we will try to use them to extract some representation of the subtitles. So we selected two models, GLOVE and GPT-2. GLOVE is a simple model which creates a representation for each word, a vector, encoding co-occurrences of this word with all the other words of the vocabulary, depending on the fact that they might occur together. So words occurring together will have representation that are closest to one another, while words that are far away, that have never occur together, will have representation really different. And so basically, this is a non-contextual model, meaning that for each word, you have a vectorized representation, and it does not change. So if a given word appear in different contexts, it will still be the same representation. GPT-2 is a more complex tool, like described. It's a contextual model, meaning that, for example, the representation for a given word will be based on the representation of the word before. Okay, so now we'll load the model in the notebooks. So, you should be at the third.
Il est en train de se télécharger sûrement.
Yes, I managed to install it and all of that, but now I'm getting an issue when I'm loading the data. And it just says it's an empty folder. I tried
Did you download this?
I didn't download it. I thought it was but yeah, but I thought like from the script. This is the stimuli. This is the other one. Right? Okay. Oh, yeah.
From search data model import all.
You already downloaded the models?
Yes.
Then you loaded the models?
I see. Like you have to navigate here.
Because I already use other like before.
But this is that search the.
No, the research portion was
En même temps, les gens ils se disent que c'est un Mac, ils arrivent et ils sont
Yeah,
il a abandonné la c'est vrai.
We try. Oh.
C'est ça qu'on avait dit sur les lancers tout à l'heure.
That's.
Il faut les 10 minutes pour charger, mais je crois que c'est C'est combien de temps la garantie maintenant? Tout va bien, c'est juste long et fatigant.
Ouais, non, c'est le problème qu'il y a au départ, c'est vrai que ça rajoute un peu.
Mais oui, vraiment.
Ça va se faire.
On est qu'à 5 minutes, quoi. Ça va, c'est pas trop stressant.
C'est pas les problèmes les plus compliqués.
T'as les gens qui sont les mecs qui sont
Ils sont aimés, ils crèvent de froid aussi.
Oui, non, mais ça,
des
par contre,
problèmes.
c'est insupportable. Hein. Parce que t'as pas besoin de mettre du froid déjà dehors à l'intérieur, tu vois. C'est un peu une logique. Surtout ici, les personnes âgées, on est tous morts de chaud en hiver. Non, mais on est vraiment dans un hôpital. Sinon, on n'a pas, eux.
Non, mais c'est parce que les personnes âgées, elles n'aiment pas.
Voilà. Ça dépend lesquels. Ceux qui sont en état de marcher, de temps en temps, on les croise. Mais c'est vrai qu'ils passent juste en dessous. Alors, on essaie de les tuer.
Sur l'autre côté.
Il était un demi-froid. Toi, t'as sauté le chat? Laquelle? C'est vraiment une classe archi sautée. Mais en gros, c'est vraiment trop. Non, c'est en bas. Est-ce que vous avez la maternelle? Ouais, t'as 8 ans en CE. Oui, non, mais c'est après la maternelle, vous avez la primaire. Là, c'est la troisième classe. Mais c'est on a cédé sur le ouais,
I can also try.
mais
We can just check where you are.
j'ai inventé non, moi, je pense qu'il a l'air de bien se passer.
No, managed to get it. Yes. To where we get to know the real loading models.
Everything.
Yes, so I need to just learn how do I get it? Oh, so maybe URL. It's able to be done in this. Yeah, yeah.
Well done. How do you feel about losing?
What? Oh, we lost. There's not much to say. But come on, France didn't play that.
I thought, yeah.
Yeah, with the post, the required with the header post just before you go. And then came I think in English like better. You can actually look at the stuff. I think actually then we want to go to Google first.
Okay.
And then you go to stats. Short. Come on, 16 to 8. That only tells you 8 to 5 on goal. 8 to 5. The session that is.
Penalty and you would just like
Tu vois que c'est parce que c'est lui ici que
More corners.
les participants.
Non, je vais continuer à parler là.
Okay.
Okay, everyone.
Come on.
So,
My possession, everything was better. Even the number.
so you should have downloaded the glove model by now.
I mean, I think I'm going to take that.
So what it does, like I described it, it associates to each word a vector of size 300, which encode information about the word and the context in which this word occurs. So you download it with this part of the code. Then you will build the glove model, which is a dictionary with the word as a key and the representation as a value. Therefore, for example, if you look at the representation of the word the, you will have a vector of size 300. What we do is that we'll go through each word that appears in the subtitles, and for each word, we'll retrieve the representation. And this will form the final matrix that we use to predict brain data. So in the end, we have this kind of matrix with one column per feature, so one column per dimension of the embedding representation, and one line per word. So we can compute it in one with one function here, but I went through all the detail before so that you can see how it looks like. Then, when extracting the representation with GPT-2, what we do is we have to do more processing for the input. It's more complicated. We do not just give the words like we did independently, like we did for glove. We need to construct a sentence, then give the sentence to GPT-2. But there are a lot of details. For example, GPT-2 has what we call a tokenizer, which might divide a word into several tokens. So it learns to segment the vocabulary, not in words, but in tokens. And therefore, a given word might be split into subparts. And then, in our case, we need to identify the tokens we need to identify how a given word might be split into several tokens. And then in the end, if we have a word that is split, for example, in 3 tokens, what we do is that to construct the representation of the final word, we'll just average the representation of its subparts. And so it's quite a lot of ugly coding. For example, this is a sentence that you want to give that is being projected onto the screen in the fMRI machine. And this is a sentence that you want to give to your GPT-2 in order to extract the representation. You tokenize it with a tokenizer, and this is the list of tokens that you obtain. So you see that at the beginning of every new word, it adds a strange symbol, a G with a dot on it, in order to recognize the beginning of a token or of a new token, or just the token that is part of a bigger word. What we do once we have this token is to learn the mapping between a word and the associated token. So this is a dictionary with all the numbers that you see. So it tells you that, for example, the word 28 is associated with a single token, which is the token index 35. And for example, the word 0, the first one, is associated with 2 tokens, the first and second one. Then there are other details that you want to take into account. For example, if you, like I told you before, the representation of a word depends on the previous context. So you can imagine easily that the first word will have no context to be integrated into its representation, while the last word will have all the sentences as context. So you understand that there is a discrepancy between the information that can be leveraged by the first words of the input sentence on the last word of the input sentence. So here we design the input to the model in order to have globally the same amount of context for each word. So given a sentence, so each token is converted into an ID, a single integer that represents the just which token we're talking about. What we do is that we will use a window, how do you say it? A sliding window with a step of one token. So imagine a sentence. I will first look at the first 100 tokens. I will give that as input, and I will retrieve the activation, the embedding, the representation of the model for all the tokens. Then I will move one token on the right. I will give this sentence to the model, and I will retrieve only the last token of the sentence because I already have retrieved the previous tokens, and this enables me to retrieve the activation of the last token with like 99 tokens of context. And I do the same for all the words. So this is why it's quite, it takes a lot of time. But in the end, as you retrieve the activation only for the last word, you know that each word for which you retrieve the activation adds the same context to build its representation. And therefore, you have less discrepancy when representing each word of the input. So in the end, what you give to the input is this kind of batch with one line for each word for which you want to retrieve the activation, and several X tokens of context and one tokens for the word for which you want to retrieve the activation. And then we also retrieve the index of the word for which we want to retrieve the activation. So these are complicated details, but that you don't want to maybe remember for now. In the end, we have, like for glove, a data frame with one line per word and one column per features. Meaning, if you model build 768 representation of each word, you have 768 columns. And so what we do after is the encoding paradigm that Pravish presented to you. So there is no much difference, except that contrary to the kind of processing that you might have done when working at the pixel level, where you have a direct match between the number of features, number of feature which is equal to the number of image and the number of scan. Here, you have a matrix with the size number of words in the subtitles, and your fMRI data as a size number of scans. And because you acquire one scan every 2 seconds, or every 1 second and 49 in the French case scenario. And during this 1 second and a half, you have like 6 to 7 words that occurs. And therefore, you need to downsample your data. And there are several methods that could be done, like Pravish presented you. What we do in this repository is the convolution with the href kernel. So you've seen before that if you present a Dirac stimuli, which is a stimuli which has no duration and which is quite short, this will generate a specific baud response. And this is a kind of standard response with which you want to convolve every features from your from your matrix, from your predictors. And once you've done this preprocessing, you are ready to align your convolved matrix with the fMRI data. And so this is done with the encoder class. I don't know if you use already some class in Python. Mm-hmm. And so you can access the code directly, and you can interact with it, play with it. So that, yes?
Is the href function that you would use to convolve this response to stimuli, does that differ across brain regions?
We use the SPM baud response, the standard SPM response that is being encoded in Nylon library. And basically, it's the same across all brain region. But I don't think there's so much differences between that and using a finite impulse response model. See, there are so many factors that in this in the huge bubble of infinite dimension, it's not so important. Will be the cross validation in your data. Could be the number of splits that you use, how you split your data, all the pre-processing that you do to your features. For example, imagine you're selecting the representation. Imagine a model which has several layers, like GPT-2. GPT-2 does not normalize the output of its layer. It normalizes it at the entry of the layer. Therefore, if you retrieve the output of a given layer, they might not have the same amplitude. And if you just concatenate this kind of representation and give them to the model without rescaling the layers independently, then this will decrease greatly your performance. In this code, I implemented a simple ridge cross-validated from scikit-learn, and I observed that it has poorer performance than another ridge that I encoded myself, because you do not control exactly for the number of splits, how the split is done. So it's difficult to have the end on all these parameters. And therefore, there is a lot of custom design that you need to do with these encoding models. And basically, in the end, so I don't know how long we have for the rest of the presentation. But there are three, at this stage, there are three main parts. So the encoder, which is a linear model that we're going to use to fit the model array representation to the fMRI data. There is the fMRI pipeline, which is a pipeline to process my fMRI data. So you can do dimension reduction if you want, scaling, and so on. And the feature pipeline, which also do scaling and dimension reduction. So what you do is that you do a cross-validation, and you train your data, you train your encoding model on some training data, and you evaluate it on the rest of the data. And in the end, once you train your model, you can visualize the I don't have the updated maps. Oops. You can visualize the final Pearson correlation. So sorry, there are some holes in these figures. Shouldn't have some holes because I didn't use the right masker. And so in the masker that I use in this first version, it removes some of the interesting voxel at the periphery of the brain, and this is why you have some holes. But if you run it correctly, you shouldn't have these holes, because the masker comes from another set of data. And basically, this is the kind of data that you have. So you see mainly activation and frontal regions, as well as around the STS, temporal and temporoparietal regions. I can go with you through the notebooks to have a look at all the details, if you want.